In this experiment, we're going to explore the oxidation states of manganese using color to differentiate them. As we do each week, we'll emphasize aspects of lab safety and continue wearing appropriate PPE while working in the lab. We will explore oxidation reduction or redox reactions using transition metal chemistry and color changes by applying Beer's law. Redox reactions are reactions in which the transfer of electrons occur. For the transfer of electrons to occur, we have to have two things happening. A substance has to give up electrons and another has to accept them. This means for each redox reactions, we have two halves, the oxidation half and the reduction half. These two halves will always occur together. Chemists use oxidation states to keep track of the electrons moving during a chemical reaction. As with many things we do in chemistry, there is a set of rules that we generally follow to help us keep track of electron movement. You will need to know these rules and follow them when you're assigning oxidation states in redox reactions. Let's look at an example. What is the oxidation states of the following atoms in the reaction? We have the balanced chemical reaction. Iron metal reacts with oxygen gas to form iron 3 oxide. We can see that we have oxygen metal, which is in its elemental state, so its oxidation state is 0. Oxygen gas is a diatomic species in its elemental form, so it's also going to have an oxidation state of 0. Now, if we look at iron 3 oxide, this is a compound with an overall charge of 0. Following our rules for oxidation states, we know that the oxygen atom has a tendency to have a -2 charge in compounds, so we're going to start with that being a -2. There are 3 oxygen atoms, so that means we've got 3 -2 charges. And again, all of this has to add up to 0. So whatever our oxidation state is for iron is going to need to balance that. A quick bit of algebra tells us then that the oxidation state for iron has to be a +3. A quick word about terminology with redox reactions. Chemists talk a lot about the oxidizing agent and the reducing agent. These words refer only to the reactants in the reaction, the substance that is doing the acting. The oxidizing agent is the substance that accepts electrons and is thus being reduced, while the reducing agent is the reactant that gave the electrons, so it's being oxidized. If we look back at our example with the iron 3 oxide, we know that the oxidation states of all the atoms, 0, 0, +3, and a -2. We can then follow how the electrons are moving, so the iron goes from a 0 to a +3. We know that's a loss of electrons, so that is being oxidized and is then the reducing agent. The oxygen is going from a 0 to a -2, so it gains electrons. So it's being reduced and is then the oxidizing agent. In this experiment, we're going to be using our knowledge of color and oxidation states to determine which DNA monomer is the best oxidizing or reducing agent. Remember, the human eye perceives all mixtures of colors. When all colors are present, we see white light. When one is missing, we see its complement. That is the one that's located across from it on a color wheel. So when red wavelength photons are absorbed, we see the color green, its complement. Or when yellow photons are removed, we would see purple. Because of the electronic structure of transition metals, their ions are able to absorb light at various wavelengths we can detect with our eyes. Not only can we detect them with our eyes, we can also use techniques like colorimetry to determine their absorbance and relate that to the solution's concentration using Beer's law. Beer's law simply states that the absorbance is related to the concentration, giving some molar absorptivity, which is specific to the substance, and a particular wavelength across a set distance known as the path length. That's typically about 1 centimeter for a standard cuvette that the light is going to have to travel through. Manganese can be found in any number of oxidation states, resulting in different colored solutions. In potassium permanganate, for example, manganese is found in a +7 oxidation state, its highest. Crystals of potassium permanganate are so darkly purple they often appear black. In an aqueous solution, potassium permanganate forms a purple or magenta colored solution. You can see this in the image of the beaker. Towards the back of the beaker, where some of the solution is splashed up on the sides, is that magenta color when it's away from the rest of the solution. The green manganate ion in the +6 oxidation state is only stable in basic solutions. The +5 oxidation state produces a yellow or sometimes blue color, depending on the pH, while the +4 oxidation state often forms a clear solution with a brown precipitate. The +3 state will form in a slightly basic solution a peach color, while the +2 a clear solution without a precipitate. So in lab, what we're going to do is we're going to set up an experiment and test a series of known solutions that we know what they are and see how it behaves. And we're going to compare that with a control solution and an unknown. One of the things to be aware of with this lab is that contamination is going to be an issue. So you need to make sure that you wash your glassware thoroughly, but both before and after use. Make sure that you never reuse any pipettes, and you definitely don't use one pipette and then put it into the next solution or the next container. Also make sure that you're very careful when you're dispensing reagents that you don't touch the side of any kind of container with that pipette tip, because that's going to contaminate your solution and our stock solutions as well. As we are working with solutions containing transition metals, please make sure you pour all of your waste in the waste containers in the hood. As always, check and make sure that the lid to the waste container is open before you pull in your solution. If you need any kind of help or have questions, please make sure to speak with a TA in the General Chemistry Learning Center or ask the lab supervisor prior to you coming to lab. Remember, if you have any questions, please ask them on the Course Piazza site. Reach out to your TA or myself via email or office hours to ask.